For the first time in history, we're holding the instruction manual for life itself. We can read it, we can test for specific instructions, and now we can actually rewrite the code. This stuff isn't science fiction anymore, it's the reality that we're navigating right now. So, this incredible power to edit our own DNA, it brings up maybe one of the biggest questions of our time, right? Just because we can do this, does that mean we should? This is the central challenge we're going to explore, looking at these unbelievable technologies and the really tough choices that come along with them. So where do we even begin with all this? Well, let's start at the very beginning, with how life itself is made. We're going to look at technologies that are completely changing what it even means to create a family. You've probably heard of cloning, but how does it actually work? It's pretty fascinating. First, you take a donor egg. Scientists carefully pull out its nucleus, that's the genetic control center. Then, they take the nucleus from an adult cell, like say a skin cell, and pop it into that now empty egg. With a little jolt of electricity, the egg is tricked into dividing, creating an embryo that can then be implanted. This has been done with lots of animals, but never ever with a human.
And there's a very, very good reason for that. Cloned animals often have serious genetic problems and tend to live shorter lives. The risk of that happening in a human is just considered way too high, and the ethical questions are so huge that it's been outlawed pretty much everywhere.
Okay, but it is really important not to get cloning mixed up with another groundbreaking technology, mitochondrial transfer. They sound similar, but their goals are completely different. Cloning is about making a genetic copy. Mitochondrial transfer, on the other hand, is a medical procedure. It's designed to stop a mother from passing on a specific kind of hereditary disease. So how does this work? Well, you can think of it like a tiny, microscopic transplant. A healthy donor gives an egg, and its nucleus gets removed. Then, you take the mother's nucleus, which has all her main DNA, and place it into that donor egg, which has healthy mitochondria. This new egg is then fertilized by the father's sperm, just like normal. And the result? It's a healthy child who, incredibly, has 3 genetic parents. You have the nuclear DNA from mom and dad, which determines almost all of their traits, but you also have the healthy mitochondrial DNA from the donor. It's a truly remarkable way to stop devastating diseases in their tracks. Okay, so we've talked about creating life. Now let's shift gears and talk about modifying it. This brings us to the revolutionary world of gene therapy. The idea that we can not just prevent diseases, but maybe cure them by going in and rewriting our DNA. The road to gene therapy has been, well, it's been really tough. Early attempts back in the 90s and early 2000s had some tragic results that honestly set the whole field back for years. But science doesn't give up. By 2015, we saw a huge breakthrough when a dog was successfully treated for muscular dystrophy. And now, today, small clinical trials are finally starting to show some real encouraging success. So what's driving all this new optimism? It's a technology you've probably heard about, CRISPR. The absolute best way to think about it is like a pair of molecular scissors that also has a GPS. It can search through our billions of letters of DNA, find one very specific spot, make a precise cut, and even replace a faulty gene with a healthy one. It's basically a search and replace tool for our own biology. How cool is that? So let's imagine how this could be used to wipe out a disease right from the start. The whole thing would begin with an embryo created through IVF. Then, at the single-cell stage, the CRISPR editing tool is injected. It goes in, finds the target gene, say the one for Huntington's disease, and corrects it. The edited embryo then just develops normally and is implanted. This has already been done successfully in primate models, which takes it from just a theory to a very real possibility. But what if there was a way to get similar results, but without editing an embryo, which, let's be honest, comes with a lot of ethical baggage? Well, that's where the incredible potential of stem cells comes into play.
Okay, there are 2 main types you need to know about. First, you have embryonic stem cells. They come from an early-stage embryo and they're totipotent. Think of them as the ultimate master key that can become any cell in the entire body. But then you have this newer discovery, induced pluripotent stem cells, or iPSCs, which totally sidesteps the ethical debate. Scientists can take a regular adult skin cell and reprogram it back to a powerful stem cell-like state. These are pluripotent, more like a super versatile toolkit that can build many different kinds of cells, like blood, or neurons, or muscle. So all this amazing technology, it gives us a ton of information. But, knowing something and actually knowing what to do with it, those are 2 completely different things. And this brings us to the really personal dilemmas that families and doctors are now facing every single day. And this quote from a physician
Wow, it just gets right to the heart of the problem. If you're talking about a disease that might not even show up for decades, like Huntington's, what is the right call? Do you test your child and then carry that heavy knowledge for them? Or do you let them grow up and make that deeply personal choice for themselves? There is just no easy answer here. And to make things even more complicated, there's the question of how reliable these tests really are. I mean, you'd think that if a genetic test comes back positive, that's it, right? Case closed, you have the disease. But what if I told you it's not nearly that simple?
All right, let's take a look at a real-world example, a cystic fibrosis test. There are 3 key numbers to see here. Prevalence is just how common the disease is. Sensitivity, that's how often the test correctly spots it if you have it. And specificity is how often it correctly says you don't have it. They all look super high, right? I mean, 99.9%, that sounds practically perfect.
But, and this is where it gets really counterintuitive, when you do the math and factor in how rare the disease is, the picture changes completely. Even with that positive test result, the actual chance of having cystic fibrosis is only about 26%. So what's going on? Well, it turns out that even a super accurate test will have a few false positives. And when a disease is really rare, the number of healthy people who get a false positive can actually be bigger than the number of sick people who get a correct one. That means there's still a nearly 75% chance you don't have the disease. It's a huge gap between what we think positive means and the statistical reality. So, when you take all of this in, 3-parent embryos, genome editing, the mind-bending math of genetic testing, it forces us to take a step back and ask some really, really big questions about the future we're all building together.
So here's one to chew on. When we use technologies like IVF to get around infertility, are we unintentionally bypassing natural selection? And could that, maybe, lead to more reproductive challenges for future generations? It's a question that makes you think about our long-term impact.
And if we zoom out even further, we have to ask about priorities. In a world with so many pressing needs and limited resources, is pouring time and money into these incredibly advanced technologies really the wisest path forward? These are the big societal debates we need to be having. At the end of the day, we're in this absolutely remarkable position. We have unlocked the fundamental code of life. Science has handed us the pen, but the story, well, that's not written yet. The final and honestly the most important question isn't for scientists, it's for all of us. How are we going to choose to write that next chapter?